As a CEO and veterinarian scientist, how do you balance the dual worlds of business leadership and the deep scientific innovation?
Hmm. I have co-founders who are deeply scientifically involved.
Okay.
No, it's an excellent question, and I don't know the answer for sole founders, and there are a lot of them, and hats off to them because it feels like a very difficult job.
Welcome to Life Science Insights podcast. I'm Sendil Karnanidhi, and today's episode we are exploring women in science, leadership, and a translational impact and Fauna Bio's journey. Our guest for today is Ashley Zehnder, CEO of Fauna Bio, a company focused on novel therapeutics based on multi-omics understanding of the drivers of hibernation across species. Welcome to the show, Ashley.
Yeah, thanks. I'm excited to be here.
Okay. Thank you for being here. It's an honor to feature your perspective on the show. Can you start by telling us a bit about your background?
Sure. So I have a little bit of a non-traditional background for a lot of biotech CEOs. Before starting Fauna Bio, I went to school as a veterinarian. So I'm went to school and studied exotic animal medicine as a specialty within veterinary medicine. Did an exotic animal residency at UC Davis. Actually became boarded in avian medicine, so that was my clinical specialty. But during that residency, I really became really interested in why different animals present with different diseases the way that they do, why certain tumors, for example, are more aggressive in some species than others, and didn't really have the molecular biology or the bench science training to explore the biology that drove those clinical differences. And so I was interested in doing a PhD to really understand more about the genetics behind those diversity and disease traits we saw across species. So I came down to Stanford to do a cancer biology PhD, really mostly focused on human cancer biology, but dabbling a bit in animal cancers and really gained an appreciation for how powerful genes that are conserved over hundreds of millions of years of evolution are in the course of disease. A lot of the really powerful cancer drivers, for example, are genes that haven't changed across hundreds of millions of years. And so really started then moving on from my PhD work into my postdoc, working more directly on the intersection of animal and disease and human health, working with some very large medical records repositories, getting clinical and from clinical data from veterinary medical records and human medical records and trying to put those together. And that's really where I met my two co-founders, and I'm sure we'll talk a little bit more about that as well.
Sure. You had a fascinating journey from veterinary science to leading a cutting-edge biotech startup. So, what inspired you to explore the intersection of animal biology and human health?
No, it's really if ending up at Stanford, to be honest. I was originally planning on staying at UC Davis and doing my PhD at the veterinary school there. And I kind of out of the blue got an invitation from Stanford as a veterinarian to join a PhD program at Stanford. They had a grant program specifically for veterinarians who wanted to do PhDs, and
Okay.
Stanford was one of the two non-vet schools in the country that had that program, was Stanford and MIT. And so I was like, well, okay, I'll go down and interview. I ended up really liking the professors and the colleagues that I met there and joining that program. But it really opened my eyes to this lack of transparency between people who are working on human biology and thinking about human disease development and really not understanding that a lot of other species have those same diseases. And so there wasn't a lot of crosstalk, even between the veterinarians, for example, that were working at Stanford or really working in lab animal medicine, but there wasn't a lot of cross species awareness in terms of what we can learn. And so it was a place where there were five or six veterinarians in the same kind of PhD grant program, and we all experienced the same thing, whether we were working in immunology or cancer biology or neurology, that there was this lack of communication about what we can learn from humans to animals and animals to humans. And so realized that that was actually a really big problem that needed to be solved, and that became really a fascinating place to try and work.
Can you take us back to the moment that led to the founding of Fauna Bio?
Yeah, it's actually a pretty acute moment. So Linda Goodman and Katie Grayback and I were all in the same postdoc lab at Stanford. This was in Carlos Bustamante's work in Department of Biomedical Data Science at Stanford. And there were a bunch of people there working on really interesting data projects, as you can imagine, genomics data, clinical data, using cutting edge at that point, machine learning and some AI algorithms to analyze that data. And Linda had always worked on the genomic side. Her background is in human genomics. and had moved more into comparative genomics, had always wanted to go to vet school. I always, as a veterinarian, wanted to understand more about genomics. And so Linda and I started collaborating almost from the minute she joined the lab on projects linking the clinical data I was working with the genomics data she was working with. And we realized that there was a lot of really interesting data coming out of non-traditional animal models because sequencing costs had gotten a lot cheaper and the data was just much more available, but it was sitting in academic literature,
Okay.
and it wasn't being analyzed in such a way that it could be easily then translated into insights to impact human health. And so how do we sort of bring this data out of where it's being generated in these really isolated academic labs, pull it together, and make it useful for drug discovery? And so we bounced around a lot of ideas of how to do that. But the question of how do you build a comparative genomics company is kind of a hard place to start. If you could sequence any animal in the world, where do you start?
Okay.
And so we had talked with our third co-founder, Katie Grebeck, almost initially more as a user, right? She was somebody who had studied human genomics, but then had really focused on hibernation for her PhD work, particularly around one species, the 13-lined ground squirrel, which
Okay.
is a really well-characterized model of hibernation in the US. And so we talked with her, like, as a researcher who works with one of these amazing model organisms, what would really advance your research? And so we were talking with her about that. And she was going out and doing some faculty job searches and actually went to do a workshop with NASA on how you can leverage hibernation biology to help humans travel in space, which is another whole thing we could talk about. Um, but NASA basically said, well, that's really cool, but we don't really have money to fund that research. So when you solve human hibernation, come back and then we'll talk to you. And she came back really frustrated from that workshop, like marched into a conference room and was basically like, look, if you guys are starting a company, I'm in. And we're like, oh, okay. Now we have a species to start with a wealth of biological data and an expert in that species who built a biobank of samples. Great. We have a place to start to prove out the process, and then we can expand from there. So when Katie threw down with us, then we had a place to start the company, and that's really when we started.
The Fauna Bio mission revolves around the translating insights from the hibernating species into human therapeutics. Can you explain in simple terms how hibernation biology can lead to breakthroughs in human medicine?
Yeah, so there's two different aspects to hibernation biology, as I like to think about it in my very simple way. One is the question of how animals go into hibernation and why
Okay.
they do that and what drives that process. And that's kind of one interesting biological question. What's more interesting therapeutically is then what happens to this little mammal body as it's going through what is actually a very punishing hibernation cycle. This is a 100-gram mammal, just like you or I, that takes its body temperature down to 4 degrees Celsius, so almost freezing, stays
Okay.
there for 2 weeks at a time. Heart rate is 5 beats per minute. Metabolism is 1% of normal. Neurons in its brain are fully retracted. It's clinically brain dead.
No
But
issues.
then every 2 weeks, it rapidly rewarms back up to normal body temperature, and that happens in about an hour.
Okay.
So that rapid rewarming is some kind of similar to what happens after a human heart attack or stroke. When you go to the ER and you get your heart attack or your stroke unblocked, you get a rush of warm oxygenated blood into the heart or the brain. Here it's happening in every major organ in the body. And so that itself is actually quite damaging. And so it's like having a mini heart attack or stroke every 2 weeks over 6 months. The body has to know how to heal itself from that damage. So then evolutionarily, these animals have really adapted ways to heal damage to tissues in multiple parts in the body. So then we can look at a tissue by tissue. Let's take example, the heart. Um, Take samples from the ground squirrel heart at time points in that hibernation cycle when we know repair is happening. When
Okay.
we look at a gene expression, what's happening to those genes? Are they going up? Are they going down? And then how does that compare to, say, humans with heart failure? So we'll look at RNA-seq studies from models of heart failure, dog and pig studies. We'll look at samples from humans that have had MI. Um, where do we see genes going up or and say a hibernator that's protected from damage? Maybe it's going down in humans with heart failure. So we start to build up this pattern of a protected gene signature versus a damaging gene signature. And that allows us on a network level to prioritize genetic networks where we see this opposite regulation. And those oftentimes are networks that contain really useful drug targets because they've been sort of proven evolutionarily that they're protective because they're functioning in a species that is getting less damage than we would. And then we validate that in various other ways, but at the core process, that's how we map those together.
Okay. But how do you leverage the multiomics data like genomics, transcriptomics, and proteomics to uncover therapeutic targets?
Yeah, they all build a pyramid. So the pyramid is really based on a foundation of genomics.
Okay.
So to do that, we work with a group called Xenomia at the Broad Institute.
Okay.
They put together a collection of 452 aligned mammal genomes.
Okay.
I think the Broad had a couple of supercomputers working on that alignment for a little over a year, so we let them do that. But what it does is, and there was a large set of publications that came out in April of 2023 in Science that walk through all the insights from that Xenomia project, which is a really great read for anybody who's interested.
Okay.
But then it gives us at a single base pair resolution how conserved is every base pair in the genome across all these 452 species. So that allows us to really quickly look at genetic networks that we find in, say, a protected ground squirrel. How similar are those networks, not just between ground squirrels and humans, but broadly across mammals? And part of the work that came out of Xenomia showed that if you have mutations in genes that are highly conserved, they're more likely to lead to disease. They're doing something important, right? Nature has selected them for a reason. So then we can look at these more highly conserved genes and say, okay, what are they doing in disease? And so that's the foundation. Then the transcriptomic piece comes in where we say, okay, we know these genes are important, they're highly conserved, that's good. But now, what are those highly conserved genes doing in a species that's protected from damage or that is damaged? And what are the differences between those? And that's actually trickier because then you need to get samples from a heart at a precise time point that's damaged as opposed to taking a blood sample. Um, and so that's where the tissue samples and biobanks come in. So Katie, our CSO, had built a biobank of samples during her PhD work that we in-licensed when we started Fauna. Katie and I actually drove it across the country in a U-Haul after bio in 2019.
Okay.
We were listening to the Michelle Obama autobiography on audiobook all the way across the country.
Oh,
But that was one of our foundational data sets where we had tissue samples from, I think, 26 different tissues at 13 different time points in the
okay.
ground squirrel that we could start to sequence.
Hmm.
Initially, bulk RNA-seq, moving more into single nucleus RNA-seq. And then the proteomics piece comes in terms of, well, RNA-seq is one thing, but not all RNA becomes protein.
Hmm.
And proteins levels don't always map to RNA levels, right? There's a different level of regulation there. So being able to then do proteomics and say, okay, we have a predicted protein, but is it actually translated and how long does it stay around and is it still functional? So all those things really stack up. We can
Stuck
also
up.
do things like epigenomics, so like
Okay.
ATAC-seq and other types of methylation studies,
Hmm.
lipidomics, metabolomics, all are kind of a layer of functional genomics on top of that.
Okay. But what are some of the biggest scientific or technical challenges in studying these non-traditional model species?
Yeah, the tricky part oftentimes is that there aren't as many reagents for these species as there are for humans, mice, rat, traditional model organisms. So if you need antibodies to isolate a protein, if you need to do a sorted cell study, you may not have a great antibody for a particular cell type that you want.
Okay.
So those are the kinds of things that sometimes are a little bit trickier. Making sure you have a good transcriptome. We actually spent quite a bit of time improving the transcriptomic analysis. We're doing another round of that actually right now.
Okay.
So understanding how well annotated are the genes from this particular species? Do we know what they are? Do we know what they do? Do we know how those genes map to human genes? So making sure that the not only the genomic reference, but the transcriptomic references are in good shape. And then, like I said, sometimes the reagents are not as available. Sometimes species are highly endangered. And so, for example, there are lemurs that hibernate very similarly to ground squirrels. They all happen to be pretty endangered as well. So we can't sample them nearly as thoroughly as we can a ground squirrel, which is an unprotected species. I wouldn't say unprotected, but they're very commonly available throughout the Midwest United States and not considered endangered. They're actually considered a pest species.
Hmm.
So there's a difference in terms of how accessible species are. Certain marine mammals, very interesting to study, very hard to sample.
Okay.
So there's always this intersection of interesting biology, disease resistance, but can we get access to the species and can we get access to the samples we need?
Yes.
So those are some of the challenges that we deal with.
Challenges.
Yeah.
Okay. And the fauna bio is pioneering the idea that nature already holds the solutions to many human diseases. Can you share some example of how discoveries in animal biology are being translated into potential treatments for human conditions?
Yeah, there's actually the three ones I like to pull out for this are pretty good ones. So, the very original GLP-1 drugs, so exenatide was the first but marketed GLP-1, that actually
Okay.
came from looking at Gila monster venom.
Okay.
So looking at these extended peptides that exist in species like the Gila monster.
Okay.
Some of our very first cardiovascular drugs, so drugs to
Mm.
decrease blood pressure called ACE inhibitors,
Okay.
also came from pit viper venom. Venom has a lot of bioactive molecules, as it turns out. So those are two very successful and very potent drugs that have been used for decades in clinical medicine.
Mm.
One that most people don't know about, because a lot of people know about those examples, but I learned about when I was doing my cancer biology PhD, is that we figured out the very basic idea that mutations in genes can actually give you cancer, not actually by studying human tumors, but by studying chicken tumors and studying
Okay.
viruses that give chicken tumors, because what they were doing is moving around chicken genes and putting them in weird locations. And that ended up giving the chickens cancer. But nobody knew that mutations in genes could actually cause cancer until they studied these naturally occurring chicken tumors of all things. And even I did a paper on this during my cancer biology PhD, and everybody in the cancer bio program was really surprised that that was the case. So some of these, I said, very fundamental understanding of, human quote unquote biology, we actually discovered in other species.
Okay. Are there any particular therapeutic areas that fauna bias work is showing the most promising today?
Yeah, we're working a lot in cardiometabolic disease. So our lead program, Fawn 1083, is a program aimed at heart failure with preserved ejection fraction. It's a kind of heart failure in patients that increases both with age and with obesity. And so it's been increasing in prevalence really all around the world. 3 million people roughly in the US have this type of heart failure, roughly around 30 million around the world. And that's a program that came directly from this earlier biology we were talking about, looking at cardiac protection. So, exactly as I mentioned before, looking at genetic changes that are happening in the heart when it's protected from damage, and then mapping that to compound signatures and being able to find tool compounds that then we can optimize to develop novel IP and novel chemistry that fauna can then own. So then based on that work, we have a lead program that's just entering IND-enabling studies that we hope to put in the clinic later next year. But that's based directly on hibernation biology. The second area that we work on in cardiometabolic disease is the metabolic component. We have a large discovery collaboration with Eli Lilly to find novel targets for obesity. And
Okay.
that's again based on metabolic flexibility and changes in energy expenditure that we see in the ground squirrel during hibernation. And
Okay.
so all of that work is being done as well, but it's really looking at more of the energy expenditure component of hibernation.
A lot of studies are going on, right?
Yeah, exactly. A lot of things. Yeah.
How do you envision fauna bio contributing to One Health, improving health outcomes across both humans and animals?
Yeah, it's a great question. And actually, I tried really hard to start a One Health group when I was at Stanford. It didn't really take hold after I left, unfortunately, but it made me a little bit sad. Um, But yes, I think part of the kind of higher level mission of fauna is to make really people aware of just how similar we are to a lot of the animals that we share this planet with. We like to think of humans as being particularly unique, and we certainly are in certain ways.
Mm.
But at the end of the day, we are mammals that evolved from an evolutionary process, and our biology is driven by the similar evolutionary processes of our bodies adapting to change and environmental change. And so we can learn a lot from how other species have adapted to similar types of environmental changes. And so a lot of what we would like to do is show that there is a way to derive really novel and valuable insights to improve human health by looking at these other species
Mm.
and drive more attention and research and care around looking at these species in their natural environments and
Okay.
not just thinking about animals as a vessel to try to mimic a human disease, which is how we kind of think about mice and rat. They're easy to keep in the lab, and we can manipulate their genes, or we can give them certain drugs to make them mimic a certain human disease.
Mm.
We actually want to appreciate the value of the biology of these animals in their natural context. Where
Okay.
have they evolved to suit their environment? Can we learn from that to ultimately improve human health?
Okay. Interesting. As a CEO and veterinarian scientist, how do you balance the dual worlds of business leadership and the deep scientific innovation?
Hmm. I have co-founders who are deeply scientifically involved.
Okay.
No, it's an excellent question, and I don't know the answer for sole founders, and there are a lot of them, and hats off to them because it feels like a very difficult job.
Okay.
I'm fortunate enough that I have two scientific co-founders who both I understand hibernation very deeply and understand genomic analysis very deeply. And so
Mm.
both of them are able to both build and run a
Building.
scientific platform and also inform the science behind all the projects that we do. So I
Okay.
can then focus more on the external facing parts of the business.
Okay.
So the secret is that it's a founding team and not just me.
Not me. Yeah. It's a teamwork, right?
Yeah, for sure.
What have been some of the key lessons learned while scaling a company built on such a novel scientific foundation?
Yeah, it's been really interesting to see what kind of people thrive at Fauna. When we started out, we were very small. We were 5 or 6 people, really for the first 3 years. Yeah, 3 years. And then we went up to 15 or so pretty quickly.
Okay.
And it was interesting to see who really came in as an individual contributor, but couldn't really scale into leading a small team. And that's something that a lot of small companies have to deal with. But we also have people who are particularly mission driven. So people who have sort of come to us a little bit out of the woodwork and have said things like, I didn't know there was a company doing what you're doing, but you're the only company I want to work for. Do you
Okay.
have a job for me? I'm like,
Okay.
well, what do you do?
Yeah.
So we've had folks like that. But we've had an extremely stable group of people with very little turnover because people, I think, are really driven by the really unique approach that we have. We have a very strong culture. I think part of that culture comes from the fact that we are a triad in terms of our leadership. And so the fact that myself, Katie, and Linda worked together so well as a founding team and have for 7 years.
Mm.
That trickles down into the teams that we lead that are all highly collaborative and we're also work together very well. So it's a highly cohesive team, sort of very low ego. We all are just very focused on getting the work done and supporting each other to do that. And I think that lends itself into having really highly motivated and committed folks working with you.
How do you foster the culture of curiosity and scientific exploration within your team?
Oh, it's hard because everybody wants to do lots of cool science, and not all cool science leads to commercial impact. So, what I try to do is give people a little bit of flexibility to explore side projects because you never know where the next innovations are going to come from. So, 5, 10% of someone's work might be like a small discovery side project that is maybe not directly related to the thing that we're working on. But typically, then, like for a good example is actually we have a little internal team that's called the Dream Team. It's discovery, research, and emerging animal models. And
Okay.
what they do is they go to academic conferences, they talk to academics, they think about what are the next emerging models that are coming out. That group identified a few years ago now the spiny mouse as an area, an animal that we wanted to start working with. It's a non-hibernator, but they can heal multiple tissues in the body without any scarring or fibrosis. So it's a really interesting model, had a lot of support behind it, and we initiated a collaboration with the University of Florida to look at brain inflammation and also skin healing in the spiny mouse. And so part of my challenge to that team then was, look, we can do a pilot project with the spiny mouse, but then I need to see things moving into my pipeline, right, eventually before we can do another one. Like I can't do another discovery collaboration until we see a target or an asset or a molecule from the spiny mouse, is something that I can actually promote as part of our pipeline. I think it's just taking a stepwise approach and making sure that the projects that you're working on are directed toward the company goal, but giving a little bit of flexibility so people can explore a little bit on the edges.
Yeah.
But it's always a balance of that. And there have been some folks who said, I really want to work on this thing. And it's like, well, we're really not doing that thing.
Okay.
And so then they've left and moved on to where they can work on those things. So there's a balance of those things, of course.
Yes, that's true. That's very important.
Yeah.
Uh, I saw you are featured as a part of Women Who Lead in Life Sciences. How do you see the role of women evolving in biotech leadership, and what advice would you give to emerging women scientists and founders?
Yeah, no, it's a great question. There's been a lot more support, I think, for women leading biotechnology companies, particularly in the last, I would say, handful of years. There's actually a really great community now. It's called the Biotech CEO Sisterhood. I didn't start that group. I'm just a part of that group that supports female CEOs in biotechnology, and they're becoming more and more organized. It was a WhatsApp chat, then it was a Slack channel. Now it's a 501(c)(3). So there are more organized ways that people are supporting each other in terms of advice on careers and salaries and referrals and networks and investor intros and all of those things. But then more casually, a lot of us do mentoring as well. So I mentor earlier stage founders pretty frequently.
Okay.
People who are just getting their company starting, wanting advice on how to do that and how to network and how to meet investors. And then we experimented with a program earlier, actually just a few months ago, with a group called Young Women in Bio that works with middle school and high school students
Mm.
and exposing them to opportunities in biotechnology. And so hats off to one of our research scientists, Evelyn Tran, who really organized that effort for us. But we had 20 high school students come in and work with Evelyn and her team to do some basic tissue staining and some basic genomic analysis and learn about and then see the impact of that. Like we're staining for this protein, but what does this protein mean in the context of disease and how does it impact the work that we do?
Mm.
And it was really nice to see some light bulbs go on in terms of, yeah, going from talking about the science to then actually being able to do science on the bench. And that was actually really fun. And hopefully we inspired a couple of those people to think about a career in science where maybe they hadn't otherwise. You never know. But it really scales from kids in school all the way up
Right.
through people who are starting companies. Yeah.
Yeah, and moreover, I feel the mentorship is very important, both in personal and professional journey.
Yeah, of course.
Okay. Looking forward, what excites you to most about the next chapter for Fauna Bio?
Yeah, I think for us, it's really seeing our therapeutics impact patients, right? I think we've been in a
Mm.
discovery phase now for several years. We started from nothing. We started from raw data, samples in a freezer, right? And so
Mm.
building the initial data sets, building the platform, having not one, but two pharma partnerships, selecting our lead program, optimizing the chemistry, filing the patents, doing all of the in vivo work, getting to a development candidate, and now embarking on getting IND enabling studies done for that lead program, and then seeing that hopefully enter the clinic. I think that's going to be really a validation of all of that path to science to see patients improving in their outcomes because of a drug that we found using our unique approach is really, really where we hope to go, hopefully very soon, but not, knock on wood.
Okay. Good luck for your journey.
Yes, thank
Um,
you.
If you could collaborate with any organization or research group in the world to accelerate Fauna Bio's mission, what would it be? One, why?
Oh my goodness, it's such a good question. I think there are so many interesting groups around the world that are collaborating on high resolution and well phenotyped human genomics data. I think one example we've talked to more recently is some of the folks in the UAE who are generating data around the Emirati Genome Project and getting really high quality human genomes and really high quality clinical phenotypes. I think whenever we can correlate our data with strong human genetics, it always makes our job of translation much, much easier. And so as we develop a more diverse genetic data sets in humans where it's not largely white Europeans, which is where a lot of the genomics data around the world is skewed, our job becomes easier in terms of figuring out what are the right human diseases and populations for us to work in. And so anybody who's got a large amount of that human data, it's really interesting to think about collaborating on that.
Okay, that's interesting. Uh, what are the daily habits or mindsets that help you to stay open to discovery?
Goodness. I try to make time to meditate occasionally. I
Yes.
take time to ride my horse a couple of times a week, and it sounds a little bit maybe frivolous, but I will say that my mind is a lot more calm and open when I'm riding than when I'm sitting in front of my computer. So I do get insights a lot of times when I take time to actually close my computer and step away, than
Okay,
trying to, do everything on your desk all the time. So
that's true.
taking time away from that work environment and kind of getting out. I actually get a lot of stimulation from traveling.
Traveling.
Travel quite a bit. I'm usually going somewhere once a month. Um, just getting into new environments, meeting new people, going to conferences, having conversations with people you didn't expect, oftentimes will make connections that you don't expect to. And so I find that quite stimulating as well.
Okay, nice. Finally, the last question. If you had to describe Fauna Bio's journey so far in one word, what would it be and why?
I think it's probably a word that's similar for a lot of biotech startups. I think it's been a bit of a roller coaster. I think we didn't know where we were going to go when we started. We had this wonderful idea, and we didn't, know how well it would work or if it would work. And then it's been really rewarding to see, things progress and start to finally take off. I think there's just been a little bit of wandering in terms of figuring out which indication for our lead program and what are the right data sets and what are the right types of pharma partnerships we want to do and learning how to build teams in the right way. And I feel like we've sorted a lot of those issues out, and now we have a really high functioning team. And then, of course, that's on the backdrop of a market that's also been doing this along the side. So just learning how to navigate those natural ups and downs. The biotech markets themselves are often a bit of a roller coaster, as people know.
Okay.
So being able to be lean, be able to pivot, and be able to focus the company in a way that matches what people are looking to do. Like right now, cardiometabolic disease is a great place to be. It wasn't 3 years ago. Um, so sometimes that can change pretty quickly.
Okay. So yeah, that's all we have for today. And thanks for joining us today. Hope you enjoyed the conversation. It was truly interesting and insightful discussion. Thank you again for your time and sharing your valuable insight and expertise with us.
Yeah, great. It was a great conversation. I appreciate the time as well.